Hello, and welcome to another episode of the Way We Grow podcast. I am your host, Nick Wolfe. My co hosts today are Gabby Staten Island and Cosmic. Our guest today is Jesse with Mothership Labs, where they specialize in tissue culture, breeding, compliant SOPs, and training. Their mission is to support the cannabis industry through science, technology, while still aiming to preserve the culture and genetics that started it all. So Jesse, welcome to the show. How are you doing today?
I do great. Thank you for having me on today. This is checking out the show. It's excited about what you guys are doing and glad to be a part of it.
For sure. Well, we were talking a little bit before the show, and you were kind of explaining a few of the different things that you have going on currently. So, I think one of the first thing our viewers are going to notice, and to any of the listeners out there, check us out on YouTube. But you have a huge amount of plants sitting behind you. So why don't you talk a little bit about like what you're doing today and what's new in your grow world?
Yeah, so I'm down in Florida right now, working with a company. They do a lot of ornamentals and all kinds of different plants. So this is their TC lab. They produce a crazy amount. And what we're trying to do is help, build a system to where you could benefit breeders, cultivators, have stock of just disease-free plants, and kind of build a new model essentially. So I hope we'll be able to tell you more about that as it develops. But a lot of good people, fantastic growers, and kind of shifting into being able to help preserve and provide at a much larger scale. So uh, definitely been traveling a lot. I was just in Michigan too, and thankfully I'm in Florida, so I'm humid again. But yeah, it's pretty cool to see like so many plants and the management and everything, the operations behind it. It is a ton of tissue culture here. So
For sure. It definitely looks like it. And I think you were saying that this facility alone produces over 5 million unique explants per year.
I believe so. And they do, I mean, they have like 200 acres, huge facilities,
Wow.
a lot of different plants, nursery, and that's kind of what I see, in the future, that at least in some areas, starting with genetic stock and professional breeding, kind of evolving into that area. So yeah, lots of plants here. Um, so it's exciting to see things at a large scale and what it could be. Big part of our goal is like, how does that apply to the industry, and how can we kind of shape that so it benefits growers and breeders that kind of started this and have been, doing this for a long time.
Yeah, I feel like that's really important. It seems like there are a lot of breeders from yesteryear, to use a term, that still don't have access to tissue culture or different lab testings that would help them make either more educated breeding practices or decisions, as well as also just being able to preserve a bunch more genetics at a lower cost, considering costs aren't going down when it comes to
Yeah,
overhead in the cannabis industry. Where tissue culture, though, as an investment, could definitely lower your overhead when it comes to even just something as simple as maintaining a large stock population.
absolutely. Yep. And why we came out as tissue culture and try to at least educate and help support that side is, I've been growing and around cannabis since I was like 10 years old, I think, growing up in Kentucky. I started there and then went to commercial operations and then to biotech and then back to cannabis. So I had that mentality like I'm not sending my breeding lines to somebody else in a lab, you
For
know.
sure.
Um, so that's kind of how it started, is like teach people how to do it and make it easier, better, more affordable. And then we can start trading and do a lot more business that way. Like I could send stuff to your lab. We can, trade phenotypes for breeding and compound that kind of success. Um,
Absolutely.
so yeah,
Collaboration for the win.
collaboration, man. Yeah.
Yeah.
So front facing, it's tissue culture today. We see huge need in the market for that. We do a lot of, molecular breeding and operation stuff, and just trying to support where we saw the needs for cultivators in the market, and just build off that,
For sure. Well, you kind of already touched on it a little bit, but let's take it back a little bit and talk about your first experience with growing plants in general. What got you into wanting to pursue a career or a life path in plants? I mean, it sounded like you did cannabis pretty darn early, but was there anything that kind of piqued your interests earlier than that?
I feel like it's always somebody's older brother, that makes the introduction. I think that's what it was. And just being young, I was like, this seems cool, and being a part of that group. And I wasn't like smoking a bunch of weed that young. I tried it. Just being around the plants is what interested me. So Kentucky, the culture there for cannabis has been going on for a long time. I mean, there's people in the mountains that have been just generations of growers there. Like that's what they do. That's what they have been doing. Um, so it's definitely been around for a long time there. And as I got older, I think I was looking more in the scientific side of things that I was just interested. Like, how does the world work and how does this plant work? Um, at the time, it was like a cool plant that I could attach science to. In school, it's like corn and wheat. And I was like, I don't care about corn and wheat other than eating it.
Yeah.
So I think that's what did. I was like, I can be cool and learn science behind weed. I kind of started it out. Um, Growing up, as I got older, my father ended up getting neurological disease and things like that. At the same time that you started getting indications that cannabis can help, Alzheimer's and all these other diseases. So that stuck with me early on, and where I wanted to continue pursuing the plant and the research side. So a lot of stuff in between there, getting older and going through, high school and everything else. But um, then friends started getting cancer or disease that it could potentially help cure or alleviate. So um, outside of growing around the country and doing everything on tour, we'll say. Um, I was like, I have to learn how to grow it. I have to learn how to manage operations. I have to learn how to breed, and then I have to learn the science. And that's kind of where I started. So I started learning how to grow from, older brothers and everyone else in those communities. And then moved out to Colorado. That's where I wanted to, be like, okay, this is a professional environment now. And started there as a trimmer, actually.
How old
So
were you when you moved out to Colorado?
it doesn't seem that long ago. It was 2014, so I would think I was like 28, I want to say. Wow, it's crazy to think about.
Yeah.
Um, and at the time, because I was like, you know, hustling, doing everything else, and just being in that side of the industry. To get an apartment, I had to get a job and show everything on paper, and I was like, oh man. So moved here and was delivering pizzas to start out. And I was like, whatever it takes. So I was highly motivated. I was like, I got to make change in my life to pursue this stuff, right? Um, a lot of good people in my life supported me down that road, and so going from that world to dropping a large pepperoni off for like a $2 tip was just crazy. I was like, whatever, I'll do it. And then like, I'll take it, man. Like, this is what we got to do. So
How
um,
long had you been growing before you moved to Colorado?
when starting at 10, I did grow here and there and play around with it. I think getting serious with it, I was closer to 16, 17, where I was like, I'm going to grow weed. Um, and there's a lot of different ways out there. By then, indoor, was taken off. We were just used to outdoor.
Hmm.
Kentucky soil is incredible. So it's really easy to grow. Like, you could flick a seed out there and it'd come back, and there's just some amazing weed. But getting the indoor game, we had, HPS lights and all that. Um, it was like a different learning curve, right? Like nutrients and all these other factors that I was like, you can't just throw it out there and it grows. So started getting on forums and learning from other growers that I knew, and so yeah, I've been growing for a long time. A bunch of different methods. We had some hydro guys in town. They're like, "That's awesome weed. I like this one." So it's huge community out there that I got to get experience in genetics and start breeding early on and learn some of those things before research and a lot of the science behind it started coming out too. So um,
Well, talk a little bit about some of those early growing experiences and kind of where you found success and where you might have had some trepidations that might have led you away from a specific style into another one.
yeah, so Kentucky is high humidity, right? Uh, very high. It's the end of the season. Um, I remember one. I think it's Alaskan Thunder. We had like under a highway, like we just let them go. That one lasted into December, just no problem at all. So it was like the easy way of growing that like I knew. And I love outdoor uh, Kentucky soil grown too, which is phenomenal. We had some stuff up in the hills there that was like perfect all year round. And I got into the indoor stuff, and that's where I was like, I don't know if I like this at all. Just way too much management and measuring chemical fertilizers and all that stuff. Just produce a different product I was unfamiliar with because I didn't know how to do it at that, starting out. Um, but really, when I started talking to like older growers that had been doing it a long time and learned that I could grow some like really, really good weed, just follow what they're doing. Uh, that's when I started enjoying it a lot more, because if you're growing bad weed, it's not fun. So the
Yeah.
getting over that hump, I guess. And there's, few different people. There weren't huge grows. I was growing up around Lexington, so you have outskirts of town and some of those areas that were easy to set up, some basements and then we had farmland away from the city that we could go out to grow a lot more. So I think the genetics there was the most exciting part, at least looking back. A lot of old school stuff I haven't seen in forever, but the stuff the generational farmers had didn't really have names for it was the best. Um, if you're thinking selecting over years and years of this, some of these have resistances to that
Hmm.
high humidity, botrytis and stuff like that, that grow just incredibly well in those areas. And that's how they fed their families, and that's how I was taught. Like, this is part of our family's heritage and how we're going to survive. So that was kind of beaten into my head early on. I was like, just don't mess with people's genetics. Don't mess with them. Everybody had their main cash crop that was, a Northern Lights cross over here, and product was just traded because like I want some of that. I'll give you some of my Northern Lights, and Afghanis came in, and then hybrids, and I think that's what got me on breeding and um, trying to learn more about it because it's hard to identify new genetics when you didn't know that world existed. Like everything is weed at some point, and then somebody brings you something special. Like, what is this? Like, where did it come from?
Yeah.
Um,
When you
so
first found that super special weed, since you were kind of already plugged into the growing community in the area, did you immediately kind of go, "Oh man, how are they growing it? I want to learn how to do that because this stuff
absolutely,
is the best." And what were
yeah.
some of those like methods that you saw that people were doing that, in your opinion, produced the best bud when you were first finding some of that stuff that you thought were levels above the other stuff?
It was the soil grown. Um, at the time, it wasn't like all these inputs and knowledge behind what they do is like those little tricks that these old guys had. They're like, yeah, just let it sit there for a while. I think the one method that was actually discussed, so you remember subcool mixing in the little mini pools and stuff, cook your own soil. started doing some of those recipes and modifications to it. And that's where I was like, oh man, this is like crazy weed. Before you see a lot of that, like call it flat weed back then. Like it's green, tastes phenomenal, everyone loves it. But in comparison, it's like it's just flat weed. Like it gets the job done. Um, and you had these new mixes coming out from some of these known breeders, and people started catching on. I was like, what is this? It doesn't even look like weed to me. The taste, the just everything was just incredible. So I think that's when the organic, like it was all organic at that point. The weed that I was smoking and getting older brothers and their friends bringing stuff in from Cali. It was like, this is insane. But I kind of gravitated towards just those organic mixes and how they're keeping that soil alive. And they didn't know the microbial thing, there's everything behind it. They're following a recipe too.
Right.
Um, so learning that way it was awesome because it was familiar to me from growing outdoor. I'm like, yeah, just nature takes care of most of that, right? Um, so it's definitely stuck with me. Nowadays, I want to go back and grow more outdoor, but so I just gotta. I'm growing them in tubes and stuff right now, so
Yeah. So then,
get
like, yeah,
back.
what translated you out of where you were focused on growing, and then you said, I want to actually go more into biotechnologies and find out more. It sounded like you were really interested in how these plants were growing and what was causing them to do their different things and processes. So, what kind of led you into that path?
It started because I was growing just some flat weed, right? I was like, I got to make my weed better. So you start reading and get into soil science, and then down that rabbit hole was, I would say, the first step. And then my weed started competing. They're like, "Man, this is really good." I was like, "Science is science is the way, right?" And just kept reinforcing those pieces for me. And like my dad was a researcher too, so I had this scientific mind from what I was told, and um, I was in that world of research, right? I'd
Sure.
meet other researchers when I was little, and so I had a lot of that kind of pushing me in that direction too. And I think weed being in my life at the time, I could attach it to that. Like I'm cool because I grow weed, but I also love science. I think it was important to me when I was in that early stages of my life too. Acceptance and all the things humans go through, right?
Right.
Um, and just continue to follow it. Just figuring out how things work.
It sort of adds a legitimacy to something that was seen as something so infamous in terms of what a career path could take if you wanted to follow something where you were growing cannabis, but then you started to talk about it in this very more higher education scientific terminology. Some people would take you a little bit more serious, and you're not the first person that I've heard say that.
Yeah, that's it. And I think the communication it brought to you where you're equal, right? It's like this guy grows the best weed, but I know some of these things, and it starts bridging that communication and building those relationships, I guess. So it gave me a different perspective to where like I can do all these things that still cool and you know associated and accepted, I guess. Um, and we meet every once in a while and share our experiences and our weed and learn more things and start learning together as a community. So definitely a big influence, just learning, so I can come back and be like, check this out, like look what I found out how to do. Or and it started becoming a lot more fun when the more I knew, the more fun I had meeting up with some of my friends and stuff. So
For sure.
definitely a huge influence.
And if I did my math correctly, you said you've been doing tissue culture for 12 years, but you moved out to pursue cannabis cultivation in Colorado in 2014. So you were already dabbling in tissue culture before you moved out to Colorado, is that correct?
Yeah, so I did started with mushrooms, much like a lot of other guys, right? That was fun as well. And I wasn't doing like tissue culture every day or going, hard down that rabbit hole. I came out because I was still learning about weed. I was like, I figure out how to grow this at a commercial scale. Um, so it kind of on the back burner for a little while and reintroduced by somebody else that just passing more knowledge, and uh, motivating me to start learning more about that. Just another avenue that we could develop tools with. Um, Learning, like, first when I started getting into it, I was like, this is just unfamiliar, just very different from mushroom work, you know, and but I was pointed in the right direction so I could start building the knowledge base and playing around. I had my own equipment I've had for a long time, and starting to get into the cannabis side, it was very difficult. I was like, this is not working at all. But it's getting used to the tools and figuring out what's possible, and knowing that some of these things exist in the world of science and cultivation uh, made me a better grower and more motivated to learn, I guess. But yeah, it's been definitely been around a long time.
For sure. I mean, I know that in general in agriculture, it's been used for different fruiting trees and stuff since around the 70s in high production, but was even dabbled in the 30s and 40s. So
Yeah,
it's got a
absolutely.
long history, almost a century of use within agriculture, which is super
Definitely.
impressive for something that is still almost viewed as the cutting edge of new technology in a blossoming market like the cannabis industry.
Oh yeah, yeah. It's for hard to propagate species, right? I think it
Right.
started with orchids and um, yeah, coming out and everyone's like, "Have you heard of tissue culture?" It's been around for a long time. It's hasn't been perfected, I feel, for cannabis specifically, and the applications and long-term storage and all the other stuff you can do with tissue culture.
I
So
think
that's
I think part of
oh,
the
go ahead.
reason for that is that cannabis is not a difficult to propagate plant. And
Yeah,
if it was difficult to propagate, then we probably would have gone to tissue culture much sooner. But because it's pretty easy to
absolutely.
take clones,
Yep.
we haven't done that. I have a quick question and a comment for you. First, the question is, are you familiar with the grow stores um, New Earth in Kentucky? They were in
New Earth. I
Yeah, they
feel
were
like
in
I
Lexington and
have.
now they're
Yeah.
in Louisville.
Yeah. Yeah. Okay.
Yeah, so there's some
Did they?
go ahead.
I think so. Yeah.
Yeah, so they had grow stores in the 80s during Green Merchant, and they still
Hmm.
have one today in Louisville. And we're actually interviewing them tomorrow on the Your Weed Stories.
Oh, that's
So I
awesome,
was
man.
wondering if you
Yeah.
knew those guys.
We might know some of the same people. So the kind of like grow the hydro shops and stuff there. Obviously, that's where people went to get like Fox Farm and
Right.
all the supplies. But some of them, I haven't kept up with. I know some of them kind of changed names or locations because it was starting to get watched a little more and be like, all right, what's going on here? And I know one of them moved to Louisville, so I'm
Right,
wondering
that's
if
the
it's
one that
that
I'm
same
talking
that
about.
same crew.
Yeah,
Oh man, that'd
now
be
they're called One Love.
crazy.
Yeah.
Yeah,
So
that would be crazy.
yeah, we're
Yeah.
interviewing them tomorrow. And the comment that I wanted to make is that most people don't realize that Kentucky and Florida were hotbeds for growing cannabis. You know,
Oh
people
yeah.
people when they think about
Yep.
cannabis, they think about California, and but yesterday I think Nick and I or me and someone else were talking about the Gainesville Green, famous weed from
Yep.
Florida, and
Oh
so
yeah.
much cannabis was grown in Kentucky as well. It
Like
was
number
just
one
hidden.
export, I think,
Yeah.
for a while. And the weed I was used to, and California had great weed too, but like some of these Kentucky boys are crazy. Like how good some of that flower is, man. It's unbelievable.
I could
But yeah,
imagine.
definitely it's been around a long time. And there's places out there like you just don't go. You don't want to go out there at all. Um, that's kind of how I grew up, like just respecting the plant and the people. Like don't mess with it. Let them do their thing, let them keep their genetics, and everything's cool. Because it was a way of life, like they're feeding families and trade and all that for a long, long time. I think I was lucky to get brought up in that side of the community too, just to teaching me respect for all these things. So, but yeah, insane high quality weed has grown in Kentucky for sure. Um,
Nice.
yeah,
Well, tell us some of the exciting things in your grow world right now about tissue culture. Like, what have you been developing? What have you been working on? Where have you found some successes and some failures that might be able to help teach the community that's looking into either starting a small home tissue culture lab, or even commercial cultivators that may be listening or watching this that are looking into potentially sourcing good tissue culture. What are some of these different things that you feel like you could shed a little bit more light on?
well, there's a lot. There's a lot there. So tissue culture is an extremely powerful tool, right? If used properly. Um, the main pieces of that I hear or discussed about right now is like genetic storage, pathogen remediation, right? Micropropagation. So what I set out to do was I've run all the MS media, DKW, everything in between, and I
Can you
always
explain
work.
what DKW is and what MS media are for those that don't
Oh,
know?
yep. So Mirashigi and Skoog, and that is basically these names are the scientists who created them for the most part, right? Um, they discovered that they have all the components to sustain plant life in vitro. In vitro meaning, I think it's Italian in glass. So sustaining life in glass, essentially. So all the what that means is you have your nutrients, hormones, vitamins, just all these things that your cells and tissue need to survive and grow, right? In combination with light and your environment, temperatures. So if you look at the different forms of that, DKW, just another form, is driver Kawasaki walnut media. There's specific ones that are formulated for tobacco and different plants across the board that scientists work on and develop and validate. Be like, this is for best for this plant, right? Um, so starting out, the a lot of the woody culture media and protocols and everything was just used for cannabis. DKW seems to work better for growth, but it's made for walnuts. Um, so if you think of it in that lens, I was like, we're giving the wrong food to these cannabis plants, right? They want completely different profile of nutrients and vitamins and everything else. And that's what I was used to using. I was like MS, DKW, and some modifications. So when I started getting into it, we actually started doing research at a research laboratory, molecular genetics. We installed tissue culture lab. We had GCMS, HPLC, so we could look at all the terpenes and just everything for breeding. So I might have been one of the first out in Colorado that we had full laboratory setup, grow rooms on rotation, planting seed, and we were trying to identify QTL markers, so it's quantitative trait loci. So basically, like where these genes are and what are they responsible for. The example I'll use is like the color purple. It's like, okay, where do these genes that are responsible for the color purple live? Um, when we identify them, we can test for them, pop a bunch of seed this big, and tell you if it's going to be purple or not. It's like kind of the short version of that, which we can
For
get
sure.
more into that later too. Um, in order to do that, we had to sustain life and use tissue culture and develop these things. So there's kind of the background there. But starting with those available media and public research and everything we could gather, um, it starts at woody culture protocols and everything else, right? So you quickly see like, all right, we can get these to live, we can get them to root, but they're just not healthy. They're not healthy enough to manipulate And that's what I try to teach is it's not like micro propagation, and it's you want to learn how to manipulate this tissue within this environment and setup. So I think the exciting things that I've been working on just this past year is fine tuning what that is, like the exact uh, components to tissue culture media for cannabis and hemp, um, the exact hormones and just the entire program to support that. So went, I think it's the entire year, 12-hour days, just going through it. That's been the most exciting for me as far as what we developed and what we're working on now, because it makes such a huge difference. Like perfect health all the way through, rooting, no problem, subcultures, zero loss in health throughout the process, because we want to get sustainable storage, right? We're just taking clones of different genetics. If it was seed, that'd be different, right? That's um, the best way to store genetics, I say. But since we want to hold on to all these clones, we want to start there first because we're breeders, we're cultivators, everything else. So we wanted to store it long term without mutations or loss of the fidelity of the genetics themselves, adding on other stress factors that cause epigenetic changes uh, and things long term that could affect how these genes are expressing. A lot of people say genetic drift, and which don't get me started on that. It's, but
That's for a population group. It's not for a single plant.
so happy to hear you say that. Yep. Um,
This Wolf
but
guy's
yeah,
pretty smart.
yeah. Oh no, we started talking. I was like, all right, I'll be on your podcast, sir. It's great. But you look at all the issues, right? You're cloning off mother. So we want to develop the tools that actually work, right? Um, I've been doing that with MS and DKW media and everything else for a while, which is like it's just hard to use. They're not healthy. You can see deficiencies in the plant. You can see things are just harder to root when you pull them out.
What were
And
some of
on
the
the
major changes that you made from using MS or DKW media to your current setup? I feel like that's going to be something that a lot of people should hear because I have a feeling I might know a couple of them, but from someone that has as much experience as you, I feel like it would be really good to hear that.
Sure, yep. I hate to say it, but it's just more CalMag, man. We'll start there. We'll start there. Oh man, yeah. After all this time, I was like, "Are you kidding me?" No,
Was
so
it
that's one.
calcium nitrate so that you're also increasing the nitrogen load?
So lowering the amount of ammonium nitrate, right? So that definitely contributes a ton to hyperhidrosis, meaning you get that translucent look, and they kind of shrivel. Um, so yeah, there's a lot of factors there, right? So you start at ammonium nitrate, so you definitely lower that. That's like immediately available nitrogen that they can use quickly. Also has way too much ammonia, right? Um, so the other one is CalMag. Calcium, magnesium need to be raised up. I just started using calcium nitrate. So what else? Man, there's a lot. Yeah, there's some things that you I just stripped out altogether. Um, One of them, I think, cobalt chloride. DKW has nickel. Don't need nickel. Has way too much sulfur. So you have to balance sulfur. And then if you look at the how nutrients are absorbed by the tissue, right? It's through diffusion. So if you can manipulate the density of your agar, you can start controlling how much of those nutrients it's getting, or the rate of that absorption, right? So all those components together are made a huge difference. But then you look at the vitamin complex, right? A lot of people, there's Gamborg B5 vitamins, um, thiamine hydrochloride in that. There's way, 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 way too much. Because all these things, if they're not balanced, it's going to change how these nutrients are absorbed by the plant. It's going to change how the sugar is processed. So specifically thiamine hydrochloride, it will kind kind of catalyze how that's processed and pulled in. And if anything's out of balance, it's just like just picture a deficiency, right? All this
Sure.
has to be kind of balanced and done, and then proper ventilation for your vessels is huge for cannabis. Um, so DKW does a great job, or at least I use MS sometimes if I'm out of my mix. It has a lot higher nitrogen content. So if you're doing a Meristem dissection, you could start getting that process moving a little quicker, I found, um, than DKW. And getting into more tissue and growth, DKW seems to work better. The vitamins had a huge impact, though, more than I'd expect them to do, I guess. Um, so if you look at, there's, I guess, a reference called Liebig's barrel or Liebig's law of the minimums,
Yep.
right? If you're missing
Got
one
the
of those
staves. Yeah.
planks, yeah, yeah. And I could send links or pictures or whatever to help share with your viewers too. But it's the same kind of concept. And then there's molders, nutrient interactions, right? All of that stuff applies in vitro. It's still a plant. It still needs this balanced nutrition, and we're just promoting it with hormones and supplementing sugars because it can't photosynthesize and everything else, right? So if you kind of break it down like that, it's like cannabis wants cannabis food. Tons of references we can look at, like, all the nutrients that we use to just grow weed with, right? So if you
Yep.
start there with that balance, you can build and make something effective. Um, there's other amino acids and some things that I started working with that are helping to control that rate and make it more bioavailable and process that we found extremely effective. And I think that's what started making cutting time off of growth. Like we can get rapid, crazy growth from some of these final formulas. But in order to understand that and compare, we run every single trial, public research, every formula that's available just to understand and start manipulating those factors, right? Um, Deionized water, huge, or RO. I know some people use coconut water, and um, all of that is going to relate to absorption of these nutrients and being able to speed up, slow down, and control that metabolism, right? The light intensity, the light spectrum, everything. So every time I discuss tissue culture, people are like, "Give me the formula." That's not it. I can give you the formula, and you can just fail miserably still, right? You have to have the internal components and the external, and the techniques, and that's what makes it successful.
Well, that's what
So
a formula is, right? It's not just one little thing; it's the entire thing that that
yeah,
you have
yep,
to put it together correctly. I have a question based on
exactly.
something that you just said about light. And so first you
Mhm.
said that these plants can't photosynthesize, and but then you said that the light is important, and this is something that Nick and I were discussing, and also, um, I have a little hobby tissue culture lab, with a friend of
Yeah,
mine,
yeah.
and he was telling me. So my friend was saying that we don't need very much light, and
Hmm.
Nick said that in his research that you need like 150 to 200 of ppfd, and I'm looking at
Hmm.
your jars over there, and they're stacked one on top of another with what appears to be fairly low light. So if you could answer the
Yep.
question about like what are the light requirements generally, I know it's.
Absolutely. Yeah. Well, picture being able to control all this, right? So you're looking at, and it's a two-part kind of trick question, right? The photosynthesis part. So I'll start there. Photosynthesis, they're creating sugars, essentially, energy to grow the plant. We supplement sugars to help, provide and boost that, especially at the cellular level. They're not going to be making their own. A lot of species, they will stop photosynthesizing and their stomatas kind of get stuck closed, and they just become in a stasis, right? If you have the ventilated caps and CO2 still present, that's something I haven't measured. I would love to, and do some deeper research on that. But that's kind of like textbook. They slow and then stop photosynthesizing. Is it true? I mean, I'll, I'm gonna look into it. We'll see. Hope we get some answers, right? But
From From what I've heard,
oh, good.
it's they do stop photosynthesizing, but they don't stop their rubisco cycle. But they just
Yep.
supplement the photosynthates that they would produce through photosynthesis with the provided carbohydrate load within the media. So
Mhm.
they no longer need to provide their own carbohydrates to still continue this rubisco cycle of nutrient processing and continuing to build cellular growth and
Yep.
cellular multiplication.
Absolutely. Yep. So we're looking at developing sugarless media, right? And so think about that. The hybrid systems, we need CO2. You need kind of these like traditional things, plants, how we grow plants. Um, that'd be huge, just way less contamination or issues and, sterile work and stuff like that. But I think getting down to measuring it and being like, okay, this is true at maybe at these points. And what does that kind of process or curve look like
Right.
just to get a deeper understanding? So get into light intensity. Um, if you're able to like slow that metabolism and process down, right? Cooler temperatures, lower intensity, and you're just slowing all that process. Maybe higher density media, so slowing that diffusion process as well. And these are all different species behind me here. Um, when you get at scale, you don't want everything just growing rapidly. You need to control that production cycle. And you can use a combination of these things to slip, make that happen. Um, for cannabis, I think I had one in vitro, the same vessel for about a year, and pulled it out, and it was just fine. But if it didn't have a vented cap, it had really dense agar and higher concentration of the of nutrients in there, so it could last. It's just kind of slowly pulling that up. That gets you an idea how you can manipulate and change some of these factors. But my main production rack, will say, it's like 50 micromoles per square meter. That's it.
So that's very
Um,
low.
it's low, right? But it's spectrum, so it's almost balanced spectrum um, of veg and flowering, right? Your blue and red. It's almost even on there. So, and I've done, veg lights or, that heavier blue spectrum, and you can get a little more stretchier, elongated growth. But I get to the rooting phase and I wasn't seeing the results that I wanted. So, um, keeping it under the spectrum and that intensity, I think the highest I'll go is up to 80 micromoles per square meter. You're not stressing the plants out. You don't want to force them to grow using higher light and hormones specifically. You want to be very judicious using your hormones to promote that type of growth. So finding that zone where you're not just like forcing all that on there, control within a margin, that's when you start seeing health, and it gets easier because the plants they'll react faster, their tissue is just healthier, they root faster, everything else. And I've done it under, I think, 100 micromoles per square meter, different lights that I had at the time. We saw excellent growth, but it's just not nearly the same as what I've developed now, because you can get more stress that you're adding on to the on the epigenetic level, and things that we're setting up to start digging through that could affect the genetics, because in the end we want the same or even better coming out than what's going in to the tissue culture process, right? And a lot of people don't understand like how these things can accumulate and change. Epigenetic stressors and methylation can be passed on to the next generation too.
Could you
Um,
briefly describe what methylation is? I haven't heard that term used before as it pertains to like the degradation of genetics or changes in
sure,
the
yeah.
epigenetics. Yeah.
Yeah, and that's I guess an easier example. There's different forms of this that can happen. So um, your DNA is not going to change, but through stress and this happens in humans as well, which is just terrifying. You can accumulate these methyl groups. Um, I won't go too much into histone protein stuff, but it starts limiting its ability to transcribe, right? Or its ability to express. So over time, and again, it's heritable to the next generation. Over time, you can, accumulate these things. Again, it doesn't change your DNA, but it starts limiting those genes to ability to express, right? So
Sure.
when people discuss like, man, this plant just isn't the same, or it's, dudding out or whatever, it's like you've been cloning off this for 20 years. This thing's just stressed out
Right.
and it's not happy, right? So that's one form of this. And you can look in, going deeper, there's specific locations that this can happen on that starts binding it in onto histone proteins, right? That's where all this stuff is wrapped up, so where it could fit into the chromosome. Otherwise, it would just be stretched out crazy long. Um, but more of those that happen, picture like a magnet, it's just binding that to the sites and it starts restricting its ability to transcribe and therefore expressing. If I explained that
Fascinating.
decent.
No,
Yeah.
I think I understand that. It's kind of like they're almost like suppressors of expression where as they accumulate over time, you can lose more and more of the genetic expression that may have made that specific cultivar desirable initially. And one of the examples that I feel like is very fascinating within this discussion as well is I've even heard people have a specific cut and they just loved how it was performing and they're like, I just want to get this onto culture so that I can preserve it for the long run. They get it into
Mhm.
culture, they've been assured that it's safe and backed up, so they kill off their mother populations. They then get
Yep.
this tissue culture back and it has all the COAs to ensure that there was no mishandling where it could have been accidentally exchanged with another genetic. But they get
Mhm.
this genetic back, so it's for sure what they gave them, and all of a sudden it is completely different, undesirable, and
Yep.
not what they wanted to grow anymore. And it's because
Absolutely.
these different sort of genetic inhibitors that build up over time can actually create a more desirable product when it comes to the cannabis that we like to smoke and consume, as well
Mhm.
as, maybe there's a picturesque quality to it where something removes the ability for the bracts to structure in a certain way but the trichome density remains the same. So all of a sudden it's just this more frosty, resin dense flower that then when removed the flower structure becomes larger, which technically is a
Mhm.
healthier plant because it can support greater embryo life for more seeds, because that's the
Yep.
entire purpose of the plant in nature, is to produce seeds so that it can preserve its genetic lineage. So I've
Definitely.
heard this,
Yep.
be viewed as, oh man, it's ruined this cultivar that I love, versus almost now we have to say, listen, we want to get the cultivar that we love on tissue culture as soon as possible because we will be able to go back to that same expression time
Mhm.
and time again without viral loads, pathogen loads, other sort of genetic stressors that sort of are sitting there, like you said, and they attach themselves to the plant's
Yep.
genetic structure over time, where it's only then removed through either like a meristematic tissue culture where you're removing it from the vascular structure and all these different
Mhm.
things. I
Definitely.
find that really fascinating that you brought that up. Thank you.
Yeah, because if you think about, oh, do you have a question, Gabby?
Oh, what I was going to say is this is something that has been experienced with, I think most pronounced is with the sour diesel. And so,
Oh, yes, the
I
sour diesel.
I
Yep.
have a clone that me and my friends have had for decades, but it's just not the same. It's gone
Yep.
through those epigenetic changes, and
Mhm.
like It's just it's what it is. I think in another podcast what we talked about is, if you have a clone of, let's say, Wolf, for example, and one of the clone, you have two clones, right? And one of them eats a healthy diet every day, and then the other one eats Burger King and Taco Bell. By the time that
Yeah.
they're, 30 years down the line, they're going to be significantly different. And then if we take the
Definitely.
clone of the Taco Bell wolf, it's not going to be the same as the clone of the wolf that ate a healthy diet all of his life. So,
Absolutely.
so the lessons
Yep.
are, number one, always eat a healthy diet, stay away from Taco Bell,
Eat some
and
eat some fruit,
but also,
vegetables.
like Nick said, is we want to tissue culture these genetics early on, and if we would
Yes.
have tissue
Yep.
cultured the sour, 10 or 20 years ago, we wouldn't have this problem that we do now.
So that's, and you hear using tissue culture, specifically Meristem culture, right? It resets the genetics just like it was from seed again, and that's not true, right?
Right.
It does a lot of these health, benefits, reviving, getting rid of all of these pathogens and everything else. Even
It's not
without a
going to make
virus,
you young again.
it's not gonna make. I'm
and
working on it.
and it's
Trust
not
me,
and
I'm
it's
trying.
yeah, and it's not going to take
Yeah,
away all the bad things that you did, right?
but it could, or I think it could, right? So a lot of this has to do. Um, so just a if you go down really, really small, or let's say meristem dissection, right? The majority of these issues, you have endophytes, fungus, bacteria that you have no idea are even in there, right? Over accumulating, even viruses that people just don't test for, right? So that's a huge factor that could limit vigor, yield, performance, all those things. We can get rid of all of that. And that's the mechanical action of just taking the, new cells, right? There's no tissue for those to live in. Um, but if you get to, and same with viruses or most viruses, if you get to these changes or these modifications, I guess, on the DNA itself, then how does that be removed? There's some indications to it's the NAD pathways, right? So niacin, nicotinic acid, can promote removing those. There's some, it's kind of a different mode of action, I would say, or describe it as, that some of these sites are filled up before, methylation can kind of stick on there. So it is possible. And I think some of that does get removed. Um, if you're using some of the the right formula or additives or combinations, right? And we're at the point now where we can measure these things, and technology didn't have all this before. So we have new sequencing methods. Next generation sequencing came out, right? And now we have nanopore sequencing, where I can sequence in field applications on a piece of equipment probably this big, come out to the grow, sequence it, and then run it, right? So we have the technology, and now we can accurately measure and monitor the epigenome, right? So now we can verify these things have been removed or accumulating. What we don't know yet is the ecosystem that all of these things create amongst themselves. That's where I start just going crazy. So you get methyl groups, and then ubiquitization, all these other long words, and what kind of pressures do the interactions between those cause? Like, we keep going down that rabbit hole, but the more we understand that, the more we can create and figure out to truly clean genetics down to the DNA level, right? Removing these methyl groups. So
It sounds like, if I'm understanding correctly, you're providing almost like a growth hormone or a regulator to the plant to help promote its expediting growth away from contaminated genetic material, which makes me think a lot about how resetting genetics, if you are using clonal propagation, is extremely important to do it regularly to maintain vigorous growth, because vigorous growth will help not necessarily remove pathogens, but prevent the buildup within the tissue and create a lower threshold, or a lower economic threshold that you need to meet in order to make sure that it's still maintaining at a specific output as a crop. But it sounds
Yeah.
like
Oh
what
yeah.
you're able to do is use that to then even completely remove some of these epigenetic changes over time by successively outgrowing the different aspects that are preventing a genetic expression that was desirable that is no longer found.
Yeah. Well, everything's always more complex, right? There's way more, deeper stuff you can get into. That is my hope, and that is what we're building towards. Um, I will never say I can do that until I have the data in front of me and I've proven that. But if you think these things are possible and this is how it functions, it's like, I mean, that's possible. It's like, okay, well,
For
we have a
sure.
path forward to where we could potentially, truly reset these genetics, and maybe we get the sour diesel back to where it was, right? Even in the grow room, the result is genotype by environmental interaction, right? Um, you can get these things to express more or less, and we can measure that. We can do RNA sequencing and all these, bunch of high level science stuff, and we can measure gene expression. Um, we can now see, epigenetic pressures and things that are restricting their ability to express. We know the exact, sites of gene regulation and that controls those things. It's like H3 on H3K1 or something, like a very specific point. So that's how far science has come, and how do we manipulate it, right?
It sounds
Those are
like
the
you can
things
make a
that,
very targeted approach, rather than having to sort
Mhm.
of use like a shotgun blast to try and get everything all at once, or hope that you hit a few things while you're trying this specific remediation method. You can almost now take the plant tissue, run it through the analysis. Uh, it spits out the data for you. And of course, I'm simplifying this, but it gets you the data. You
Yeah.
take that data, you go through. Oh, this data fits in here on the chart. I can now take this application or this methodology, apply it to that plant tissue, and we should have a 10% success rate of removing the issue that we found. Retest all the data,
Yep.
or retest all the tissue after
Yep.
that next successive generation, take that plant that shows that it was removed, and boom, you have the next line within your steps that
Exactly.
to remove everything that you want to remove from the epigenetic changes.
Since
Yeah. And yeah, go ahead.
I was gonna say, since I'm the simpleton here and not as smart as you and Wolf, to simplify it and use the same analogy we were using before, if we have Wolf who's been eating Taco Bell and Burger King for 30 years, there are things that he can do to improve his health. Like if he has high cholesterol,
Yep.
maybe he takes cholesterol medication and stuff like that, and he can lower it. So there's certain things that we can do, with tissue culture and other means to improve the health of our plants and our genetics, but we don't know how to turn back the clock and fix all the damage that we've done yet. Maybe someday we will. But currently, like we,
Exactly.
we know
Yep.
what we know, and we're learning more every day, and it's exciting.
Uh, Exactly. Yeah. Like we have, because there's so many fields of research that like I want to learn everything. There's a problem. I'm like, well, I'm gonna go learn this now. But if you look at how fast technology and research and everything is rapidly developing, right? Now we have AI, um, integrating those fields of research with AI, with genetics, with um, then I think we'll find solutions really fast. Um, but it's a combination, right? Keep your genetics. If I'm breeding, I'll pop a bunch of seed, put a coffee in vitro. If you're keeping it in tissue culture and it's not healthy, you're just adding more problems to it, right? So just don't keep it in tissue culture. Keep a healthy mom until you can figure it out. Because those types of you can run into mutations, epigenetic pressures, everything else rapidly if you're putting them in vitro and um, doing it that way,
If you
right?
stress something that's
So
already stressed.
exactly,
Yeah.
it's just like, just kill me, please, right? And you can always reculture it and keep it rolling, but you're just adding more of that onto it, right?
I'll always
So
refer to the first part of tissue culture as the proofing phase because you got to make sure that everything that's going into culture is healthy enough to sustain growth. So
yep,
I think that
yep,
was, we had Cosmic on earlier, but he had to jump out. But one of the things that he was talking about is the potential for contamination when using a single culture vessel for multiple explants or multiple meristematic tissue cultures, just
Mhm.
based on either the use of improper aseptic techniques, improper cleaning practices prior to putting into culture. So, what are some of the steps that you take to help reduce the or to increase the viability of your initial explants when going directly from a mother population or a stock plant population at a facility?
yeah, that's a so referred to as stage zero, right? There's a lot of preparation you can just do with your mother plant to start. If you think endophytes that are in the tissue, you can remove a lot of those. Use a product with like BioSafe, Zerotol, something like that. It's pulling it into the tissue, and it can kill a lot of these anaerobic things and knock those down quite a bit, right? Stuff, Pythium. When you get Pythium, it goes all the way up and it starts getting limp on top. If you add those, then it starts straightening it up because it's killing that in the tissue itself. So if you're having trouble with contamination, I would definitely start there. Um, Getting it, just knocking as much down as you can. When you take your samples, um, I haven't seen a huge difference with the right formulations. Always keep that in mind in health or subculture or everything where I'm taking that tissue. The best is obviously going to be lower parts of the plant. And if you think endogenous hormones, exfoliation or this photomorphogenic response, right? If it's in low light conditions, it's just going to try and stretch towards that light. There's hormones in there that are already in the tissue. So those always have a faster response as far as growth. But you're also getting thinner little stalk to cut. So we can surface sterilize as much as we want, but if it has a really thick stalk that you're trying to put um, into the agar, you're not going to penetrate and kill everything inside, right?
That's a really
So I
good
go
point.
down this. My like initiations are probably like this big. Let me see, tiny. Remove all the tissue that's not going to contribute to healthy growth. You don't need it, right? Because if you leave it on there, it could be harboring contaminants that you might not see for months. Like it'll just build, and then a leaf touches the side of a vessel, and then it's contaminated, right? So the purpose of this is like get rid of all that stuff. We know that it will grow and just be really, really healthy in culture with the formulas, things we've developed. So you start there. Get rid of the chances that there's contamination in deeper, thicker tissue. Um, it makes a huge difference.
Can I stop you
For
and ask you
sure.
a quick question?
Yeah, yeah.
So, I mean, you're kind of blowing my mind right now by saying that you should use the lower tissue on the plant because what I've always been taught, even from the people that taught me tissue culture, was that you want to use the top newest growth.
Mhm.
So are you saying that that's not true? That, you know, something on the bottom that may have been there for longer and is thinner and, stretched out is actually better parent material than like the apic, the top of the plant?
Yeah, that's what I use, and there's different sides to it, right? If you're taking a meristem, um, you get that apex of the tissue. Those are going to be bigger up top. Remember, you're cutting all that tissue off, right? Doesn't matter. They're way easier to dissect and isolate,
Because they're bigger.
and you have more aggressive kind of new cell growth, cell division and growth, right? But if you're putting in, node culture or just a shoot tip, um, I always take it from the bottom. I think University of
Wow.
Guelph came out with a paper that's saying, subcultures, they have a lower decline in health from the basal stalk if you're taking from that, which really like those low shoots that are um, just coming off the stalk on the bottom, right?
For sure.
Um,
I'm.
and
It makes me
there's
think of like UV radiation and how that can also damage genetics within any plant
absolutely,
and or any living creature. So anything that has a little bit of a more shade response, where it is trying to push a hormonal growth rather than using the light directly on it to promote and drive the growth, you're going to have a little bit of a greater potential for healthiest genetic tissue. And
definitely.
this is something that I feel like, just for a little clarification, like if you are doing clonal propagation, using a lower branch may not produce the most vigorous clone. So
Mhm.
using upper growth for clonal propagation is usually the most advantageous, simply because it does have a lot more available photosynthates directly on site ready to be used because of the
Yep,
photosynthetic process that's happening that close to the light. But when you're not
exactly. Yep.
relying on photosynthesis to provide the plant with the carbohydrates it needs for growth, then something with a higher hormonal load in it does sound like it would do a lot better in vitro.
Yeah. Yep. And you're not having to add these extra hormones and, and it makes a big difference, right? So the thin little shoots that are popping up, easy, way easier to sterilize, like thoroughly sterilize, so you're confident it's not going to start growing in, a couple months down the line.
Right.
And I see just a faster response, but that also has to do with formulations and things like that. You're going to see a dramatic difference. Um, So if you just stick to
So,
just thin tissue that hasn't been, because a lot of those top pieces that you want to take regular clones or cuttings, right? You want those big thick stems. Um, Kind of go the opposite. It doesn't feel like it makes sense, but that's where I see the very most success as far as sterilizing and preventing contamination.
that's an awesome trick for people that are trying to get into this and find success early on, because it's such a long process to go from even nodal culture to an explant that you can then put back into rotation as either a mother or a production plant.
Mhm.
That it's really hard for some people to then lose all that progress and want to continue. We even see that even just with home
It's
growers
crushing. Yeah.
popping a seed and getting
Yeah.
some, thrips or some disease or just powdery mildew or something that hinders their overall grow, and they just don't want to do it again. So limiting
Yeah.
those
Yeah.
little potentials for issue is so huge.
Yeah.
And I would like you
Definitely.
to touch on the uh, the difference between nodal and meristem tissue culture, and what you prefer to do first if you're going in to do genetic preservation. Would you start with nodal tissue culture, or would you start with meristem? And just to preface this a little bit, I've always thought that you go from meris or nodal to meristem while it's all in vitro, so that you can have you do that zero generation to really make sure that it's the healthiest tissue possible. Yeah, so it'd just be great to kind of hear your opinion on whether you like to start with nodal or meristem tissue culture and why.
Sure. So when somebody's learning, I always suggest start with nodal culture, get the process and everything down, right? When I am saving something, for long-term storage or keeping a clean copy, I always do Meristem, right? If I pop it from seed, usually I'll just do, a node, because it hasn't been exposed to all these other things and collecting endophytes and fungus and everything else. I try to do it early though, so you're not having that full xylem and things that can get in there. I'll just take one, sterilize it, keep a clean copy in. Any other genetic that I bring in, Meristem, 100%.
But do you start with meristem?
I do, yeah. So once you have a clean copy, right, from Meristem that you can multiply, everything coming out of there is clean, right? And that's the idea. I have a huge collection of genetics. I don't know where a lot of these came from, or I do, but the facility they came from, what they have, whether, sometimes I know it has a virus, and I'll take the same precautions if I know it doesn't have a virus. If you just, be safe and follow best practice and protocols, and you'll prevent the spread, right? Because you could take something out of culture you did from Meristem and just give it a virus like pretty quick, right? But new genetics, I got some of my old stuff back the other day. I just want some of the classics, Super Lemon Haze, Golden Goat, things like that, right? Whether to study or breed with, doesn't matter. Meristem dissection, that way I'm confident that it's clean and done properly. And once I have tissue in there, I can pull as many of those out as I want. The difficult part with Meristem is the length of time, maintenance, checking them, making sure it's growing. And that's something we've cut down dramatically. Like, rule of thumb, people say about 9 months. I don't want to speak too soon because I still got to go home and make sure make sure it's alive. But we could easily cut that in half, or we're looking at about 3-4 months, potentially.
Is
That
that
rate of
from
from
meristem to a
tiny little
new
cells.
like fully hardened off plant ready to go into production?
Yep.
Cool.
But that rate of growth, and this new formula just for this, I still want to make sure that's healthy rate of growth, right?
Right.
I'm still not using tons of hormones or anything, like same, kind of approach that I use. But you can have rapid growth and have mutations and other problems that you definitely can't see, right? Especially if you're breeding with it. So we're going to look further into that at a deeper level. But if you start with Meristem and it's clean, and you can feel confident in that, then everything else you do from there is going to be better than what you had, right? Because
For
even a,
sure.
a tiny little node that you can sterilize, it still could harbor like a tiny, tiny spore or something. You're not going to see the effect of it, but over time, the plant grows and those spores or contaminants grow inside as well, right? And that's the biggest piece, because the stuff I've pulled out of tissue culture from Meristem, it is explosive growth, like, and it's not too many hormones. It's because nothing else except the plant's tissue is utilizing the nutrients and everything else. Um, you push it into grow, it'll get a collection of these endophytes and other things in there, but um, that's where part of our name is the Mothership Labs. We get new sets of mother stock from this system every quarter, or however long you want, and you just don't have to worry. You get extremely vigorous, healthy plants, high yields, all the stuff that people think of when you talk about tissue culture. But you turn around, you're like, we're not seeing that. Like, why, right? So definitely start with Meristem to practice and kind of dial things in, getting familiar with everything, node culture is just a great way to burn through tissue. You can get plenty of it. Um, because once you're comfortable and you can manipulate these things and you know what to look for, then Meristem is a lot easier to like monitor. Once you get it past that first stage, it's just the same stuff like transfers, clean them up a little bit, make sure they're healthy, um, and becomes much easier. But if you start with Meristem and you haven't done this before, it's I won't say it's overwhelming. It's just like a difficult first step. You're like, I don't know if I can do
Sure.
this. The learning curve again is just practice. Like if you just get used to the motions and what you're looking for and proper technique, then the rest is easy. Well, it's pretty easy. There's some
Yeah.
troubleshooting here and there, but um, I want the stock that I grow and especially the stock that I breed with to be as clean as I can and as healthy as I can. Because talking about epigenetic pressures earlier on, right? Those are heritable to the next generation. So definitely start with as clean as you can until we get more easier solutions, right? I guess
Right.
to kind of reset these things, but which I think will come. I mean, the technology's there, which is crazy. So
For sure. Gabby, did you have a question?
I did. So, I like I said, I have a hobby tissue culture lab, and I've only done nodal tissue culture in the past,
mhm.
and I want to learn how to do meristem. And so when I went to the lab, the other day, it took so much time to prep that I didn't have time to do meristem, and I had already taken all these clones, so I wanted to stick some in nodal. If you have a clone that's in nodal and you want to do meristem off of it, would you suggest doing this sanitization procedure again?
um, I don't
Because
think
that's
you
something that
you
I was
have to.
thinking. You don't think you
Yeah.
have to?
Well, I mean, I think you're thinking the right way. That's
Well,
what we
just because
want.
you had
Like,
mentioned
make
that
sure
it's
it's clean,
possible that
right?
it's not clean when you do a nodal tissue culture, and
Yeah.
I was thinking, normally when you do nodal culture, at least the first time you stick it in, you got to sterilize it. After
Mhm.
that, if it hasn't grown anything, you just pull it out, divide it, and put it in new media. But if I'm going from nodal to meristem, do you think it's important that I then again do that sanitization process?
I don't think so. I mean, you could technically, right? You could, let's say you got a thick piece of stem, and during that process, you cut through a stem, it gets something on there and gets onto the cell tissue or the meristem you want to remove, or the tip of your tool when you're placing it onto the medium, then you could get it. But really, if you're confident it's clean, you're removing anything that those can live in anyway, right? It's not going to live in the clump of cells that you're removing. So I think you'll be fine doing that. Uh, the way I usually sterilize from tissue, so it's healthy, rapid growth that hasn't gone in vitro yet,
Mm-hmm.
and I'll sterilize those to put in. Um, but you can do it both ways. Yeah, if I have some I'm going to dissect when I get home that are just in vessels. They're huge, filling them out, but I don't have a copy that's acclimated and ready to go. I want to start the process and get the rest of my collection done, right? So you can easily pull that out under the microscope, and it's again just feeling comfortable. We have some methods that are fast, easy, and overall accurate is what you want. Um, and biggest suggestion, if you go to the dentist, ask them for some of their tools. Most of the time they'll recycle them or send them back somewhere, but they are the best for meristem dissections. You can kind of just peel those primordial structures off to expose that and just take it right off.
What type of
Um,
tools are we talking about?
so those dental picks,
Oh,
they're like the
right, yeah.
handles and they have like
The tool
hooks
that I
or different
hate.
shape. Yeah, it's a
Yeah,
tool
that's
everyone
great.
hates.
Oh
Yeah,
my God.
best thing for
Cl-
dissecting ever. Like,
Wow.
um,
All right. That's a
because when
that's a
you
gold
get in
that's
there
a
with
golden
a
tip.
knife, oh yeah, it's huge. There's some other ones we'll talk about. But you get in there with a blade, like scalpel blade, even number 11, which is like thinner. it's still hard to kind of sort through that little structure.
And it's okay. You're
And
not like ripping things off. Like
yeah,
that's
why
actually what
use
you want to
it
do is you want to
like
peel it
if
off,
these are the
not cut it.
leaves, I'll kind of like push them to the side, right? And on both sides, so it kind of like opens up. Then you can visually like see the structure and where you're going into. Because after a little bit, you're like, I don't even know what I'm cutting here. I'm just like mashing things up, right? Especially if it's on, a dissection plate. So I hold mine straight up in the air like this, and I'll get forceps, and so it's sticking up in the air, and I'll peel, the primordial leaf and any structures to the side so you can kind of see it. It's way, way easier. It's just another thing that takes practice, right? You do that 10 million times, right? And you're cruising. I can do one in like 5 minutes and just knock them out. It's all in the prep work and like I have all my tools. Everything's good. Okay. Um, because it's you get taught certain things and like humans just you're like we have to have these tools and this. It's whatever works best for you. I have a different hand shape or a different, my elbow. I got a bum elbow, and you find a tool that's really effective that works for you, um, and just use it. So I got a set of dental tools. I get them for $5 from the dentist. If you buy them new, they're like $50. You can autoclave them. Some have little tiny blades on the tip. It makes it much much easier. Um, and another
I'm gonna
one
go talk
is
to my dentist.
every time I go there, she's like, I got like five more for you. I'll give it to my clients and stuff. And I'm like, all right.
By
Um,
your clients, um,
mm,
I'm sorry to interrupt, but something that I'm super interested in is what does Mothership Labs do? Like I know that you're a tissue culture lab, but are you are you selling tissue culture clones? Are you teaching people how to do tissue culture? Are you doing
yep.
um,
All
what is are
pretty
you
much everything. Yeah.
what is it called like when you just hold all genetics like a
Genetic
repository?
preservation.
It's like a
Yeah,
bank.
yeah.
Yeah. So we I build labs. So I design, build labs, pretty much any scale. We can do fully automated robotic tissue culture labs if you want to spend the money. They exist. Um, and we started off because, I've been running big programs and breeding and research, and um, there's just a lot of bad experiences and bad business we went through in this industry, right? And I ended up working with my current partner, Jay. He's awesome. And a couple of those experiences we went through, and we were like, we work really well together. He keeps me in the lab, he says. Um, and business development, sales, he's just amazing, man. The stuff that I don't want to do, right? I'm not good at, right? And we started this because we wanted to choose people to work with that had the same values and vision that we do. So we kind of organically shaped to where we could do a ton of different things, but this is the way we can help people the most, like in the immediate term. Um, so coming out, building labs and showing people like advanced techniques and developing formulas, like here you go. Like we do all the hard work, just go. And seeing them be successful. Um, when they're on their own, now we have an affiliate lab. We can just like form a communication, right? We can send each other clean genetics whenever and build each other's catalogs and make crosses. And it's been awesome. So the long-term vision is breeding research more, more of the stuff that we love to do, and collaborate with other people that do that. We have powerful tools, and so we use all of our capabilities to create powerful tools that are easy to use and that you can apply, um, to make your help sustain the industry, because there's been a lot of collapse across the board, right? So like let's save these genetics, like preserve them. Now we can do it so they're healthy and we're not having long-term issues. We can have the real genetics stored. Um, and sometimes I'll just do it for because you run into the trust thing too, right? Like send me your genetics and I'll hold them. Um, that's why I started like building people labs, right? The first version, I just had like a mobile kit that I pieced together. I'd show up and be like, I'll show you tissue culture is not hard. Like, and you show somebody the process, and they're like, that's not that hard. Like, you can do it, man. Like, I got your back. Like, let's go.
It's funny that you
And
Very
say
cool.
that. I mean, I want to, if you guys will humor me for a second, I want to tell a little story.
yeah, yeah,
Do you know who Bill Graham is? The guy who sells the home tissue culture kit.
yeah, I've heard
Yeah.
Belgrade for sure.
So, you know, he gave me a kit, and I kept it for over a year, sealed in the box, never used it, because I was intimidated, and I just felt like I had to get ready. You know, I had to build this sterile space, and I like I said, it was intimidating. And so one day he
Yeah.
comes over to my shop because I owned the hydro store, and he comes to the shop and he says, "That's it. We're just gonna do this." And he already had media prepared, but he broke out the pressure cooker so we could make media. And then
Yeah.
it wasn't cannabis because it was my grow store, but we had scented geraniums and redwood trees, and we divided them up. We used a Rubbermaid Tough Box turned on its side with the front
Oh, no.
door of my
Yeah.
store opening. So like it was in my
Yeah.
retail space, the door was opening, and we put the plants in there, and everything grew, and we got
That's
very
it.
little,
Yeah.
if any, contamination. And then after that, I was so inspired. I did tissue culture every day. Like if you came into my store, it smelled like alcohol and bleach because I'm constantly spraying this stuff, doing the tissue culture. But
Yeah.
now I've ended up kind of in that same situation for many years where I'm scared to do Meristem. You know,
Oh,
and
I
like,
got you covered, man.
and I'm just trying to like break that seal and do it because I know once I do it, then I'll repeat, and you know, but doing it that first time, it's like it's
Well,
intimidating.
it seems
It's yeah.
so like, and honestly, it's kind of like passing it forward, right? Because where I came from is like nobody helped me for a long time. I had to like fight and scratch. You got into the industry, nobody was going to share information with you. Like that's my recipe, or I was like, all right, how am I supposed to do my job then, right? So, and we're trying to just fight against that. Be like, I'll tell you exactly how to do this and what I'm doing. I have suites of SOPs. Like, if you're successful, then I believe we'll be successful together. You know, but somebody showing me that I was like, holy shit, like I can do this. Like it isn't that
It's like
hard.
someone teaching you how to grow weed the first time
Yeah.
too. It's
Yep.
like
And it
such like
started,
how you know, and then you see it and you're like, it's not that
yeah,
hard.
yeah. And it started my entire career path. Like learning, like you need to learn in this area. I'm like, okay, cool. And just surrounding myself with people that are way smarter than myself and learning to ask the right questions because you find if you got good people around you, they that are educated in whatever subject, if you talk to them they love to share that, right? And I've been to
Yeah.
laboratories where like I've been to PhDs, like I've dedicated my entire life to this, and some of those people end up just turning out samples or having to run more business than science, and their dream when they're little kid was to be a scientist. So you start like, can you show me this or what about that? I go home and do my homework and come back, be like, all right, I ran into some more questions. Like their eyes light up. And they're like, I know all about, like he's asking about this. And that's what inspired me a long time ago to go down biotech and all this. And otherwise it kind of dies, right? You hear tissue culture lab, and the first thing is like way too expensive. You got a whole sterile room and all this stuff. And I mean, if you're doing it this scale, you want some preventatives in place, and but you don't need to do this if you're at your house. I built one at my house that's pretty crazy, but maybe another. I ended up just building a lab in my house so I could
We
do all
should we should
this.
do a lab tour
Um,
sometime of your home lab,
yeah,
just
maybe we can
over
do it.
video.
We just
Yeah.
invited ourselves to your house.
Dude, I'll just live stream. You guys just pop in whenever you want.
Yeah,
I
we
actually
want
got
to.
these. I got these AI cameras too, so we can do a lab tour and a Meristem excision too if you want.
Oh,
That'd be great.
that would be awesome. I've been
Yeah.
Yeah.
recently looking at videos on YouTube trying to like figure out how to do
Uh,
it. One thing I wanted to add is, until recently, you and maybe, and even Bill Graham and some other people, we had the guy from Genomic come on, educating people about tissue culture and cannabis. The guys that were doing it didn't want to do it. Like they want to keep
mm,
their secrets and make you think that
Hmm.
it's really hard to do because that's their business plan to like make money selling everybody the stuff. So for me, I appreciate people like you that are willing to share the knowledge and educate people. Um, like I said, it's the same about growing
absolutely, yeah.
weed. Like if we kept all the info back about how to grow weed, it just holds the entire like
Yeah,
universe back. So I just want to say thank you for
definitely.
being willing to share all this
Uh,
information, and
Absolutely,
I've
man.
been very
Yeah.
inspired already,
That's
from this
that's why we do it.
conversation.
And again, like you might find something you're like, dude, I just figured this out. We go over together and I could start using it. Like it to me and my partner Jay, it's less about like um sometimes I'll just show up at people's like, all right, I'm gonna knock these out for you. It's so free just because I love it. But it's less about gatekeeping the knowledge so I can make money and there's the money will come, man. If I'm teaching people to do this properly, we all advance in
And
this
there's plenty of
industry and we
money
can
to be
all,
made teaching people how to do it too, and building up
yeah.
labs and
Yeah, and
One.
we build a network and exchange information. It's less about me like providing mine. It's like let's exchange information and knowledge, and continue to grow.
Yeah.
So
And some of those earlier companies that did have these very proprietary tissue culture recipes for cannabis, they were evaluated at an extremely high valuation due to their recipes and methodologies. And so they were beholden to contracts to not release that information out to anybody, even if it was friends, family, somebody that was just looking to get into tissue culture, because they would void their contractual obligations and their fiduciary responsibilities to these investors, where I think one of the recipes was valuated at $10 million just for the tissue culture
crazy.
recipe. And that was, 5 to 10 years ago. And
Mhm.
that company is now out of business because they realized that you can have this awesome recipe, but if you're not then sharing it with the community and helping the community understand the value behind tissue
Yep.
culture, there is no reason to do it in the first place.
Ex exactly. Yep. And I've heard so much bad information or misinformation, right? Some of the information is like that's what they learned and were told, and which is okay. Like I understand that too. But the work we did and like the focus work, I'm so tired. I did this entire year. It's like it works. And all these things that you heard tissue culture can do is possible. And
Let's touch on one of those actually really quickly. Sorry to cut you off, but I feel
yeah,
like
yeah.
one of the biggest controversies in through the views of cannabis cultivators when it comes to tissue culture right now is the removal of hops latent viroid through meristematic
Yes.
tissue culture. And
Yep.
you and I were actually put in contact with each other through our mutual connection at Toomey Genomics,
That's
because
right. Yeah.
I had reached out to find out more about hops latent viroid remediation, because it is
Mm.
something where some people will say there's no guarantee, it can't happen, and they've been doing tissue culture for years or decades, but they're caught, I believe, in a mindset of limitations where they don't
Yeah.
think that something's possible because they just haven't been able to do it yet. And what I always like to tell
Hmm.
people, this is also for people that have been growing for 30 years, 40 years cannabis, and are stuck in a specific set of ways where they say, this is how I've always done it, this is how it works,
Yep.
and I'm not going to listen to new methodologies. I've already been doing this for 30 years. How could something be better? And so I
Yep.
feel like
Yep.
people pigeonhole themselves in a lot of these mentalities, unfortunately. But I was put in direct contact with you because of your ability that you've shown to remove hop latent viroid. And so I'd like to pick your brain a little bit about that, where you found success and how that success has sort of been able to been proven out over time.
Sure. Yeah, I mean it's a hot topic, right? And yeah, when you first reached out, I thought like a potential client, and I was like, he's asking a lot of questions there. What's going on, right? But it was great to connect. You're asking the right questions, right? Which was it's refreshing in a lot of ways. So as everything is, right, the world is it's more complex than can you remove it, can you not, right? It's different with different genetics. There is the mechanical removal part, right? Removing the cells at the apical dome, not connected to the vascular system, which a lot of these things can be moved through the tissue, right? That is one part of it, but I believe there's been some instances where they have transferred through other avenues, right? Through these cellular mechanisms, and I group that in as like more research needs to be done to be understood. But there's some genetics that are knock it out in the first try and be like, I'm the best there ever was at TC. Some genetics extremely just a pain to do that with. It's not all the same, right? So there's like the variety of how these things evolved in the tissue down to the cellular mechanisms, right? They're not all the same. Cannabis has a very different plant in some ways, right? So starting there, the methodology and treatments, and there's a variety of those available, right? You can use broad antivirals. You can do, I mean, there's a ton of things you can apply to help prevent cross contamination and have that be more successful. Rule of thumb is, I think I told you too, is like 50% of the samples, right, is what you want to expect. But there's also those, those outliers where like none of those came out clean, or all of them. So going down to the techniques, so I'm sure you guys pulled like cold treatments and, a lot of different ways to approach it. The
I actually
cells,
don't know about cold treatment. Can you briefly touch on that?
oh, sure. So you can basically I'll take a sample this big, right? Just your tissue and protect it. So I got a kind of step process to, protect it, store it in a refrigerator. And I know some people that have gone way colder than that, but it's basically really, really cold for a shorter amount of time, or refrigerator for, we can do like 2 months or so. I've adapted that into using some synthetic seed techniques as well to protect those organs and make sure we're going to get functionality out of the cells to grow. But essentially, you're just slowing everything down. It always starts with the sample that you take, right? So remember the photomorphogenic responses. You can get a plant to stretch out pretty quick using far red light, right? So why not stretch that out? It's growing really fast. Then take your explant. So already you've either limited or diluted as much as you can, right? From the bottom of that plant. Because template viral, you're talking links to this metabolism and growth. So if you can just stretch it out, and if far red light, you can get that to happen pretty quick, right? And start there, you're already ahead. So at least the viral load and everything else you're working with. Cold storage does this kind of same thing. I've heard heat, cold. It's all, I think, needs to be evaluated further to get like real truth behind some of these things. But because we're looking at just the mechanical separation, if it's not able to get into those cells, why does it matter, right? Which could
Right.
get into those cells. So a lot of that, I think, is just an attempt to kind of slow everything down so it's not spreading into the tissue you're going to just dissect anyway. So currently I just use that far red light, which you can get isolated spectrums of that. Take your explant and do a proper dissection. And that there's techniques which I'll go over when we're in the lab. I'll go over with all that stuff for you guys. So it
Cool.
makes a huge difference, right? Because if I'm sitting there smearing, HLBD all over the samples I'm plating up and so on and so forth, you got to prevent that. And that's, I think, the hardest part where you can isolate that efficiently. I use ocular scissors and, for eye surgery, things like that. You find the right tool or the best tool for the job,
So you go to
to
the
where you
eye
can
doctor and get their used tools too?
see so you're catching on, right? We just got to schedule appointments, go pick up tissue culture supplies, and we're good, man.
Alrighty, just make a list of all the doctors that we gotta grab their recyclables from, or go search in their dumpster behind
Yeah,
the building to see, like, oh man,
exactly.
we got some ocular scalpels. We need.
Well, funny enough, the last tool was like a hypodermic or hypodermic needle that you can just because it has the sharp end that you can pull that off without touching any other tissue on there, right? So
For
then you
sure.
have
Oh,
in the
that's a
lab, like,
great,
oh, I got a clean pull, and then you're able to just remove it and just kind of plate it like
Yeah,
like a
that.
sharpened
I'm
spoon.
I'm a rookie, but it's funny that you say that because I watched a video and it literally looked like he didn't cut the meristem out; he popped it out. It's
Yep.
like he just went in there and he
Yep.
went like this. Like, first he peeled it open a little bit, and then he just popped
Yeah.
it out. I was like, man, he made that look too simple. We need to do that in slow motion, or it needs to be slow motion and even zoomed in more than
Yeah.
than
We
the
can do.
microscope was showing
We'll do
because,
that.
yeah, I
It's
can't
all
wait.
technique, right? And if you think about just tissue in general, if it's waterlogged and been sitting in, you just sterilize it and you went took a break, that tissue is going to be different than, um, you picture like asparagus in an ice bath, right? It's still crispy and you can kind of snap it, right? So if it's sitting in there in your sterilization solution and it's just absorbing liquid, it's going to be a little more gummy and hard to kind of pop off, right? So a lot of those things is where you fine tune those skill sets
But
and
don't
you
you?
start.
I'm sorry, but don't you? Isn't that
Yeah.
part of the process? Like you put it in water and you soak it and clean it. Then you put it in bleach and whatever, and then you soak it in water again. Aren't they always going to be waterlogged by that point?
No, no, well, not if you're just efficient with that process, right? So and I can run. I think the most I did in one day was like 22. I would never recommend that. It was painful. But I dialed this in because it has to be efficient for people to use it, right? It goes back to that concept. Um, 50 mil tubes. I have a flat. I just pull it up, fill it to that line, add the tissue, cap it, orbital shaker for 5 minutes. And that's just with water, washing particulate and residues off for water in between 20 for polysorbate, right? Empty them out, and then I add bleach water. That's 5 minutes on the orbital shaker, another 5 minutes for me to set up and prepare to start pulling tissue, right? So that way it's not getting over sterilized or oversaturated. I'm just like empty them out, rinse, and I'm already starting to pull tissue off. So you get in a rhythm, like start with enough samples to where you get through it, and you start making it more efficient with like 50 mil tubes or like whatever you have,
So you're
right?
not sanitizing
Um,
too much at a time because then it's just sitting there in water or.
exactly. Yeah.
So
So I stick to
I
like
made that
six
mistake
diff.
the other day.
It makes huge difference,
I mean,
right?
I was only doing nodal, but still, I'm assuming,
Yeah,
you
gets all
know.
Yeah.
flimsy and yeah. So I usually cap it at like six different genetics at a time now, just because I got lots of other things I'm doing, and I can knock those out, get them in culture, be good, you know. But start with one. You can do. I use 50 mil tubes, and you're not wasting too much liquid or having to manage too much liquid, right? That's even worse because
Space.
you're juggling like big jars and space. Yeah. Um, so you can just get a 50 mil tube rack and shake it if you don't have an orbital shaker. And but by the time I get to that six sample or six cultivar, It's still good to go. If I start pushing it and do like 12 at once, the last one I'm starting to do is like getting flimsy and um, it'll still grow, but we want just the healthiest, high quality tissue going in, right?
Right.
Um, so, as I do a lot of like Lean 5S training and process procedure optimization stuff for laboratories and operations. And that's why I broke it down because it's easier to train. People pick that up easier. There's like less steps. The steps make sense. Like I'm not having to do unnecessary things. We've tested them, and you can get to the final step of dissection faster and start moving. So then you don't have a guy in having to spend all day in a lab to get 5 clean samples and throwing out everything else, right? I'm like, I haven't seen contamination in a long time, which is awesome. Just be just solid best practice and
Yep.
just follow the steps and you're good. You can just mass produce initiations, I guess,
That's awesome.
and just get fast, just the mechanics. So going back to the HLVD, a lot of those things are still valid, right? Best practice, efficiency, because if you're smearing viruses all over stuff, that's not going to do so well, right? Um, those first 2 steps are very important and have done really well as far as success. Getting that stretched out piece, hopefully that's outgrowing or diluting some of that to some extent. Um, and I've heard people say that like outgrows HLVD in culture, right? And I wouldn't be confident in that. I haven't tested it, but I wouldn't be confident. Um, and the mechanical removal, you can always put broad spectrum antibiotics, stuff like that in your media plate. Ribavirin, or I mean, there's a bunch of them you can do, but as soon as you get that growth, you're looking at surface contact, right? So you can have antibiotics, put it on there, and maybe it'll kill some when it's contacting that, but it's going to start growing unless that is getting pulled into the tissue. Like I don't see that being effective either, right?
Hmm.
Um, so yeah, I mean, it's really case by case basis, I would say. And when people ask me to do it, I'm like, I mean, this is the truth. This is what I've seen, and I'm trying to study this more at a deeper level so I can give you a better answer. But the reality is like there needs to be more research. I see on average about 50% success rate. So I do twice as many samples, depending on how many I want
fifty
coming
percent
out.
success rate is great.
Yeah. Some
Yeah.
people, that's their entire tissue culture success rate. So if you're getting that with hops latent viroid, that's crazy. That's
Oh
extremely
man,
successful.
dude, I got you
I you
covered.
know I wanted to give a analogy again, and so kind of what we're talking about is it reminds me of cancer. So for instance, with cancer, they cut it out, right? But there's still a chance that they didn't get it all out and that it hasn't, and then you got to go and do it again, and so it's the
Yeah.
same thing with tissue culture. And actually, with tissue culture, well, I don't know what they have a better chance of removing cancer or hop latent viroid, but the bottom line is you have to do the tissue culture and then you have to test and then you have to do it again.
Hmm.
And even if you test, you might have to test it again down the line or
Yeah.
because it's just
That
hard to
first
know if it's
test,
gone.
yeah.
So
I mean,
like
you can go
people think they're
undetected, right?
cured of cancer, and
Yep.
then years later it comes back because it
For
was
sure.
still inside of them somewhere.
Speaking on testing
Oh yeah.
and the length of time from your zero generation in tissue culture from Meristem, where the aim is to remove hops latent viroid. What is the furthest out you've then tested, say, a plant that has fully come out of tissue culture, is a mother plant or a stock plant at a facility that was cleaned up. They removed all their hops latent viroid plants, but they sent it out to be tissue culture. They then intook those genetics again and started following better practices to help reduce the potential for hops latent viroid.
Mm.
So, what's the longest then that you've been able to go back and test something that tested positive for hops latent before tissue culture and then tested negative out of tissue culture. Was was there ever something that you found, that after like 15 months, like there shouldn't have been a contamination problem, but it did test for hops latent again? Or have you seen that if you don't have potential for contamination, if it's not being contaminated, that it's been able to just sustain health and
Yep.
zero viral pressure after, yeah, what's that time span you've been able to see?
Absolutely. That's a great question because a lot of people assume like, here's your plant from Meristem culture. It tested negative. You're good to go. And that's not the case, right? Because of the nature of the virus itself. The plant, as it gets older and metabolism starts moving, bigger root system, it's pushing all these things around. The viral load increases. Testing in itself, the LOD, so the level of detection, it's only so low that we can identify it as the virus, right? It might not be in the piece of tissue that we took. It might, and that's why you see testing requirements like take some of this, and a lot of it's in the root system that we can test for it, right? But when they're that little, you might not notice it or detect anything at all. Okay. I might, and I'll never tell you like, put this in your clean grow. You're good, right? Because 6 months down the road, all of your plants could have it, and it'll be my fault, and I'm not gonna, I'm not that guy. So I would say 6 months, and there's people that are far more qualified to speak on this than I am that are studying these fires like to me and those. But from my knowledge and my asking people and things like that, I would say like 6 months, 8 months down the road, I would still test it. It's because I'm paranoid and I don't want it spreading, right? Um, but when it's this big, I would say, like a teen size plant, when it has a fully developed root system and it's in that final environment, I would say where, hotter temperatures, humidity, like you're gonna flower that out. I would say that's when you can feel more confident. You're like, okay, it's good to go. Um, by then, I would already have subcultures in there or new copies to where like, all right, we're clean. Then like release all those subcultures. That way you have the numbers coming back into your garden instead of just waiting for tests to come back and then you have one plant to start cloning,
For
right?
sure.
So there's strategy behind that. Not like here's your plant, it's tested in 6 months, get back to me. It's like here's your plant, I got 50 of these coming. So if we pass that test, you got 50 new moms coming or however many you want.
For
Um,
sure. How
so
how far
there's
out have you been able to test like from clonal progeny and have negative test results, or negative in the sense
um
of that it no longer shows hops latent viroid after removing it from that specific cultivar? How long have you been able to see it test negatively for?
I think the
I
opposite
mean
question
forever.
is more important, right?
Yeah.
Cool. That
Yeah,
well
because
then I
I
mean
I
he
could test it myself, right? I have qPCR, gel
right.
electrophoresis, everything. So I use that to just validate my process. Like, this isn't working. We got to change. Um, and that's what we want, right? The biggest issue is here's, people give me their favorite genetic for their brand. It was like happy to clean it up, putting it right back into a facility that's just filled with viruses, right?
Right.
Um, and I see that all the time. They're like, "Oh, son, we get vigor and all this." But like, you bite those first couple cycles, it's going to perform way better.
Hmm.
But you're not solving the issue. So
They don't have
that's why we're
proper
working.
cleaning, proper SOPs in their garden,
Yep,
and
yep.
you should always assume that you have viroid, and and
Dude, the
and do
floor is lava.
practices
Yeah,
do
that's
Hmm.
practices to not spread it. It's just
exactly.
kind of like
Yep.
safe sex with random people you don't know. Like, yeah, I mean,
Good.
Glove up.
you're not seeing herpes, but
Glove
that
up.
doesn't mean she doesn't have it,
No.
Yep,
or other STDs. So you got to be
exactly.
careful.
And
Use
Like
that's
it's
clean
that's
it's
why
scissors.
a place like this, right? And this, you see the logo. They gave me a new hat, Lab Roots. Why we are working with companies like this, we can flush your entire facility out, right? now it's a much different picture. You're like, wait, hold on. Okay. Start harvesting those rooms at like we'll schedule as these clean plants come up. We'll have a whole room ready. You harvest a room and rotate, clean it, sterilize it. Here's your first clean set. Just don't touch it. Like clean
Yep.
room style. Like don't go in there, new scissors. And as they rotate out, you're just flushing your entire facility out with your own genetics. We can do that. Um, and that's what I see the next phase of getting some of these things restored, right? It's almost like a restoration project for the industry, man.
Yeah.
Um, because there's so many good genetics that I definitely don't want to lose, because they could be studied, or they could be used for breeding, or and have these kind of varieties that are just phenomenal, man. Um, but tools like this is like we flush your whole facility out. You're way more successful, and it's almost like another chance to just test anything. Have a quarantine protocol before bringing something in your facility, right? Or just hit me up. We got some awesome genetics too.
For sure.
Um, so there's always the functional tools of tissue culture, but then there's the strategy on how to apply it so it's effective, and you're like getting the results, right? And
Right.
that's a big piece I see. Um, a lot of these places missing is like if I install a lab, you could flush out your entire facility. You can, start generating enough stock to have just run. I don't recommend micro propagation, um, unless you know exactly what you're doing and you know how to measure it, right? Um, and that I always take a chunk from Lean 5S is like DMAIC: define, analyze, improve. On define, measure, analyze, improve, control, right? And you stick to that. I apply it just to everything. I'm like doing dishes, saying it just haunts me in my dreams. But there's a lot. Oh, I see Jay poking out here.
The way you do one thing, man, the way you do everything.
Yeah. So the practical application. Like we have some of our other partners, they like group of just amazing scientists. We're on protoplast fusion labs, and we create perfect hybrids by fusing cells together. Cross species
Sure.
hybrids. We can do so
We're gonna
that
have to do another
technology.
episode.
We're going to try and reel it back in a little bit here because it sounds like we're going to
Yeah,
need to do another
let's
episode.
Yeah. Somebody here.
be done. Yeah, yeah. Oh, good. Yeah, well, I'll introduce you to Raquel. She's from this is her lab here. You want to say hi?
Hello.
Hi.
Very impressive.
Yeah, they're building some awesome stuff out here. But
Very
yeah,
cool.
we love to connect with you again. We'll get you some Meristem, dissection training, whatever, man. We'll get you in the lab
That's
remotely,
awesome.
or if you guys come to Colorado, you're always welcome. So
I appreciate that. Like I said, I
absolutely.
have to go, but
If
Jesse,
running anything I can help with, man,
super
give me a
nice
shout.
meeting you.
So
And
sounds
All right.
good, man. We'll
Thank you
do
for
it.
joining us, Gabby.
All
thank
right.
you, Nick. Thanks
Great
for
to meet
inviting
you.
me. What a great talk.
Cool. So yeah, I mean, so far we've really touched on, I feel like, some aspects of the tissue culture side of cannabis that a lot of people do find very hard to wrap their brain around, and to there's there's not an entry level tissue culture, like you kind of have to jump in. You can't just get your feet wet. You got to go all the way in.
Yeah.
And you've touched on a lot of those different aspects that really do help support, like in your mission statement, you're wanting to support what's already here within the cannabis industry and use this tool and this technology to continue that support so that people can be more successful, or find their own niche within the market that helps them succeed. Like, for instance, for the farm that I help manage, we have a huge outdoor plot, and we grow over 100 different cultivars in that outdoor plot each year. We also have an indoor space, and the indoor space only can get up to 21 to 22 harvests per year, and we're typically running only between 1 and 6 cultivars in there per year. So we can't even get through our
Okay.
full genetic library in a year. and the maintenance of mother plants and stock plants is
Expensive.
it's expensive, it's
Yep.
tedious. That's the
Yep.
thing that I find to be one of the hardest aspects of maintaining a large stock plant population is the constant need for organization, the constant
Yep.
need for proper techniques within the cloning process, that
Hmm.
if you get a hiccup, it throws your schedule off. You need to then
Yep.
retake clones. It throws off if you only have a certain amount of space. With, like, we're a small farm, we have limited space, so it's hard to maintain these different populations also with a small labor force.
Yeah.
But with
Yep.
tissue culture, it does seem as though there is a lot of opportunity in a lot of different aspects of cultivation that can kind of help bring cannabis cultivation overall a few
Hmm.
steps forward.
Yeah, definitely.
So, with your experience, and like I said before, we're going to have to come back to do another episode, especially when you're back home in your lab, because I want to go over your different breeding practices and some of those projects that you've done. I'd
That's
love
what I
to hear a
love.
little.
I love breeding stuff. So,
It's one of the, like, real fun hobby aspects of cannabis cultivation. I feel like that anybody can get into, and they can
yep.
really create something that's really unique to them. It's almost like
Mhm.
having that awesome family recipe that you share with people, and they're like, "Oh my God, that's the best food in the world. How did you make this?"
Yeah.
It's like
Yep.
we've just been doing this
Yeah.
forever.
It keeps
So,
us
like,
attached to the plant. Yeah, and
absolutely.
yep.
So, like I was saying, though, we need to do another episode, talk about your breeding, talk about your actual home growing practices currently, where you're
Absolutely.
keeping these genetics alive outside of culture, because the in vitro culture, that's one aspect of it, but you still
Mhm.
then need to have the ability to have those plants in a actual growing environment,
Yep.
ex vitro, and the different techniques and stuff that you use there in order to maintain a certain health. I feel like when you're looking at it through a scientific lens of these are either going to be for tissue culture or they're coming out of tissue culture and I need to maintain a specific amount of health, that ends up leading to really refined cultivation practices, really refined
Absolutely.
environmental lighting, irrigation practices that are
100%.
almost, they're
Yep.
so essential for actual cultivation of cannabis for flower production that
Yep.
I feel like we'd be able to teach a lot of people more when we dive back into that. But considering you've had a lot of experience in the illicit market back in the day, you were able to translate that into what sounded like helping with different medical and earlier programs within, say, Colorado and around those
Mhm.
areas. And then you also continue with your home growing and your breeding and all these different practices and aspects of the industry that you're in. I feel like you would be a great person to ask, where do you want to see the cannabis industry change or develop in the next 5 to 10 years that you feel as though we're missing currently?
Um, there's a less, there's a lot there, because I started at least professional cultivation and management and, COO, restructuring other people's programs, all that stuff. So I've seen a lot and I was able to ask a lot of these questions and get different perspectives, right? Um, I would like to see really, best practice stuff that a lot of that's coming out too, especially with the virus. And be like, if you just follow this, you keep contamination out, you keep pests and things like that to create a clean product, right? So that's one. Testing. Testing is all over the place. We had a full lab. We'd send our stuff out for compliance. We'd run the same samples on our equipment in another lab. They're all different, right? So standardizing that, because if it does become medicine, we need to know that those results are reliable and we can use it as such, right? Um, that goes also into what genetics are truly, everyone's renaming. They'll find a good cut and they rename it. How do we build around that to where it is? Oh, thank you so much. Um, how are we sure that if I go to this dispensary or wherever that evolves to, and I need medicine, how do I know it's the same plant that is helping me, right?
Right.
I think there's be application of so pharmacogenomics, right? We have the cannabis genome, we can do that. We have the human genome. Some of the groups I'm working with is to connect those pieces to see, like, we could both smoke the same joint and I'm freaking out, and you're like, I feel great, I love this. Our body, just like all these plants, are different. Our receptors, our brains are formed differently. Our experience has shaped the development of those, right? We know about the methylation and how that is an aspect of just how life um, evolves and continues to grow. So, how do we unravel all that to where we understand the application for medicinal purposes and standardize those pieces? Um, and I would also like to help, like, because we've had to create hybrid breeding programs, right? Classical methodology, I couldn't use. I couldn't grow fields for statistical analysis and because of regulation and compliance, or it was just not cost effective. How do we get better access for research and development? I know some of those are starting to evolve as well. Um, but if we're not able to grow these plants, to study them and improve without breaking the bank, um, then that side is not going to continue to evolve either, right?
Yeah.
Um, so we're all part of a groups, different groups to push these things forward and contribute. So I think if you have your own breeding program, you have unique genetics for your brand that we can fingerprint ID those at the genetic level.
Hmm.
So we know that's yours, and that adds more power to the brand and builds their customer base. And so breeding programs and more education behind how you can do this effectively in a controlled way. I can keep going, but a lot of this
No,
gets
those are
into
great.
more the molecular marker assisted selection and being able to truly pinpoint some of these things
I feel
using
like
biotechnology.
it sounds like we can wrap up a lot of what you'd like to see in the better application of the information we have available and putting those practices into use and not falling back to these old, quote unquote bro science ways of growing cannabis and producing cannabis like it's still the illicit market. Like taking this
Yeah.
and trying to truly quantify and qualify the different aspects of the cannabis plant, how it interacts with the human endocannabinoid system, and trying to use proper studies and test samples and test groups and different populations of users to help understand how things affect certain people, how we
Yeah.
can then take that data and give it to other people so that they don't have to be a guinea pig for their own medicinal needs.
Yeah. Exactly. And I talk to groups that are one has a daughter, one has a brother. Like, I have lots of friends. I've seen some that, like, they've cured their cancer. Like my best friend, he had severe lung cancer and it's completely gone.
Wow.
Why does that happen? How do we change some of these people's lives and have a positive impact from what all of us are just really good at? I know some cultivators that are far better than I am. I have a bad neck and back now because I've been growing weed for so long, but how can we just organize together and share more information? Because I feel like
Yeah.
that's a huge start right there, just being able to freely talk about what's working and how we can work together. And as a company, my partner and I are like, if we can pay the bills and just keep moving, it'll happen, right?
Yeah.
We're working on powerful tools for breeding and biotechnology and connecting these other networks. So we can at least contribute, to that, but it's
Right.
can't just be, we, so I love to see it, people just kind of like it used to be, man, like growers unite. We get,
Yeah.
and be able to make positive change because it is evolving and is going to continue to evolve and change. And it shouldn't be just the people that have a ton of money that um, is allowed, you know. So anything we can do to help for your viewers or your crews, anything.
For sure, dude.
These guys here are in the same mindset. This company, Lab Roots too. Like we're hoping to use a lot of their experience to scale and have some power behind this too. But it's still going to take work to create a model that we can keep within the community and the people.
Right.
So
I mean, that's why I call this show The Way We Grow, because it has two meanings, right? I want to teach people how to grow commercially, home growing, hobby, tissue culture, anything in between. I want to make sure that people have the tools at their disposal and the resources they need to access knowledge that has been withheld
yeah,
or gate kept for decades. But
definitely.
not only do I want to help people learn how to grow, I want to grow the community as a whole, and I want to grow the collaborative effort between different growers and people that are in cannabis and cannabis adjacent businesses. Because just like any other industry that has really taken off, it required immense amounts of collaboration between those who were working to really progress the field in a way that they thought helped the most people possible. And
Yep.
people like yourself who are out there trying to help implement best practices and remove different stigmas from practices that have been controversial or argued about ad nauseum. You're so valuable for the community and for making sure that we do continue to grow as a whole, that that's why I have this podcast. That's why I want to get people on here like yourself and get these stories told and get this information out there, because now people can reach out to you knowing that you have this background. And I keep saying it, we got to get back into another episode to talk more about your home growing, because some of the things that I do
Yeah.
like to impart the information of is, pitfalls for home growers, what you can teach new growers, where you found success in your home grow and failures in your home grow and how you learned from that. And those are all typically things that I touch on, but we're already kind of butting up on where I feel like this would be a good ending place. So I'm going to actually just kind of rein it in really quick and wrap it up with
Yeah.
a few rapid fire questions that are always really fun to hear from someone like yourself. You're a professional operating at a high level within the cannabis industry, but you still love cannabis. So what's your favorite
All
strain
right.
of all time?
Oh man, I knew that was coming too. I knew it. If I got and this is my older brother's friend, long time. I haven't seen it since, man. The Afghani and Northern Lights that he was bringing me, that was just insane.
Was that the
But
Afghuli?
the Afghani and he brought Afghani and Northern Lights, and those were
Okay.
just grown to perfection. Um, Other than that, there's some that I've been working on that they're just phenomenal because I'm breeding them kind of for me, but also as a parental line for large flowers and some of these things. But it's Mazar blueberry that I worked for a while,
Wow.
DJ Short's blueberry, and
Sure.
then a Chem 91, a bunch of Chem 91 lines and crosses. Chem 91 Skywalker OG I grew for a while, and then cross those to get hybrid vigor, and
That's
some of
really
those
cool.
are just unbelievable, man. Um,
Wow. We
Those
actually
probably be
might
some
have
of my
DJ Short
favorites.
on in a week or two.
What? Dude, I got
Yeah.
some F5 blueberry. Oh, that's awesome, man. Legend.
Damn.
Yeah.
Yeah,
Yeah.
it's such a tight knit community that it's really interesting to see how quickly things can happen when you connect with one person and how closely they might be tied to someone else that you've connected with, and you're like,
Yeah,
man,
yeah.
it's like
It's
that
amazing,
five
man. Yeah.
degrees of separation is it's only like two or three in the cannabis industry. It's really
I
fascinating.
usually hide in behind the scenes. I go out low key high tech because I just stay in the shadows, just doing, whatever. But after talking to you, I was like, man, this like I want to be a part of this because like we discussed on the phone, it's just like um, I want to use my abilities and exchange knowledge and help support, and because it changed my life, and hopefully I
For
can
sure.
least inspire, and could just change the direction of somebody's path or build into something much bigger, which that's fantastic. So,
That's awesome. Well, so favorite
yep.
strain? It sounds like you got a few. One from back in the day, the
Too
Afghan
many,
Northern
too
Lights that your
many.
buddy grew. You got some ones that you're working on now. And
Mm.
so the next question, of course, is who's your favorite breeder of all time?
Uh, that is, well, I have favorite breeders for different reasons. Like some of
Sure.
these Kentucky guys, they'll just kind of like smell the wind, and they're like, it's gonna be a good crop. Like those guys, because they have some stuff that's like doesn't even have a name. They're like, it's just weed. Like, man, this is the best weed I've ever seen. And I just love I love that style. They select what's best to them and their family,
Sure.
and there's so much meaning behind that. Um, and then other breeders, I don't know, man. It's because I've had such a huge collection for so long that I have my own breeding lines and stuff. But I would say like some of the old subcool stuff was really exciting. Just like the profile, like Vortex and some of these that I used to get back in the day. Um, and I had some of his collection left too that I need to pop.
You're the
But I
second
didn't.
person to say his vortex.
Oh, the Vortex, the baby poop smells. Oh man. Other than that, I mean, there's a lot of talented breeders that are breeding for the way that with I just picture them as isolated populations almost. And I feel like a lot of that is just bottlenecked and being changed, and that's something I have some really crazy, like different genetics that maybe we could collab and start, expanding the gene pool to get
Sure.
more unique things and stuff like that. But there's just a ton I can't even keep up with them these days. I'm just in the lab, just focusing on what I'm doing most of the time.
I feel
Um,
that.
and the guys are just making their own crosses because they're passionate. I would say that's because that's where I started. Like it's special to them, and when I'm able to smoke or grow that, I think that has the most meaning to me. It's not like a
Sure.
name or I don't care about any of that. But I care about
Yep.
the connection and the experience with the people and the plants that ties it together.
It sounds like you're a
So
fan of anybody breeding with passion and an intent in mind rather than a quick buck.
yeah, let's put some biotechnology and high level stuff behind it, and then like I said, all of a sudden, like you extract DNA once, you're like, that's not that hard,
Yeah.
and maybe they can have some of the opportunities that I was gifted. Just but that's why I got into this. That's why what excited me and what I wanted to pursue, and it just evolved, and I kept going and learning, and I was only be able to do that because I was just open and willing to learn, and continue
Nice.
learning. So I wish that upon everybody.
For sure. All right. Well, last question. What's your most memorable cannabis related experience?
Oh man, I've lived a long life of cannabis. Um, the guy that brought over the Afghani Northern Lights, right? And my friend's gonna hear this and just start rolling, right?
Good.
That was when we had the flat, we like just move this, and so you brought over this stuff. I was like, that's not weed, like that's just fake, like looks crazy. Um, and loaded up a bong. I ripped it and then stood up. So I'm like 6'2", 6'3". Blood just rushed to my head, and I like passed out, almost broke a glass table in half. That's like the one that I will never forget. It was embarrassing,
Right.
but um, so I had to be cool back then. I was like younger. I was like, I'm fine.
Yep.
I mean, there's so many memories along the way though that I'm sure I'll think of, man. Oh, and I got
I've
to.
totally passed out from a gravity bong hit before, just going from the
Oh,
bucket, standing up, hitting it, and then just standing there and being like, I held this for too long. Boop.
oh man, I was like, all right, well, I guess the other one that sticks out, which is hilarious, because I mean we're in Kentucky, right? We're some wild boys out there. I
Sure.
went to pick up weed from my buddy, and he's still in my phone, is Alligator Mike. So I go to, open the door. He had some like lizards and stuff, and we were like walking back to his living room. He's like, "Don't worry about Elvis, man. He's chill." And I was like, "There's an American alligator just walking down the hallway." It's like, what is going? He's like 5 feet long. I was like, "What is going on
What the
here, dude?"
fuck?
He's like, "He's chill." Like, you're running stuff like that, like all the time out there. You're like, "This is crazy." It was like,
But it's still just another stoner experience, right? You're just there to either pick up some weed or smoke with a buddy or something. That's too funny.
yeah, we're like, "Alligator bite." Hey, it was crazy, man. But yeah, so because it brings people together, man. It has it's done a lot of good things in my life, and I'm just science minded, and I went that direction. So that's how I try to give back.
Awesome, dude.
There's some hilarious stories along the way.
We can get
Um,
into some more on the next episode. We can even do like another little roundup. Anything else you want to touch on too? But
yeah, yeah, we do a lot of breeding stuff, advanced things. Like I love to talk about it. Just
cool.
keep me wrangled in, so I don't go too deep for your listeners. And maybe I can just send some pictures or just references, information to help people out too. Man, I love to do that.
For sure.
So
Well, Jesse, it's been a pleasure. Thank you so much for joining us today and telling your story of how you got into tissue culture, some of your background in the cannabis industry, and sort of where you want to see things go from here. You're an inspiration for a lot of people that want to get into tissue culture and are scientifically minded but want to get into the cannabis industry and have seen some barriers to entry due to the stigma and stipulations of what is required in the cannabis industry. So, again, thank you so much.
absolutely.
This has been awesome, and I really appreciate your time.
Anytime, yeah, we'll do it again. It's been a pleasure. I'm glad to be a part of this. So keep doing what you're doing. I think it will help a lot of people and bring us back together. So
For sure. Hey, that's the
that's
way we
good.
grow.
That's right. We'll talk
Cool.
soon. All
Soon.
right.